Okay, well, thank you very much. This is a great honor, and it's much appreciated. But I also want to just point out that the work that has been done in my lab by my colleagues in my lab, they really deserve this honor as well. So no, it's not working. Oh, it is working, it's just slow.
Yes.
Okay, so there are 4 topics. I just want to briefly tell you about some of the projects ongoing in my lab. But I want to spend the majority of the time talking about TBC1D24, and I'm hoping to get some good ideas from you. There are 2 projects that are unpublished. One is on small molecule therapy for TBC1D24-Associated epilepsy. And the other one is to test a hypothesis that we have about why there's so much clinical heterogeneity with the mutant alleles of TBC1D24. All right. So with regard to my lab, there are several goals. One is to identify the genes associated with novel forms of human syndromic and non-syndromic deafness, reveal the wild-type functions of these genes, characterize the pathophysiology, and develop therapies. But recently we've focused on ascertaining families with syndromic forms of deafness, and this is largely being done by my close friend and colleague, Sheikh Riazuddin in Lahore, Pakistan, where he's one of the associate deans of the medical school. I can just tell you what we're finding that's quite interesting is we're finding mutant alleles which are more damaging for the genes that we normally associate with non-syndromic deafness, which isn't surprising given our understanding of Usher syndrome. Okay. So the strategy is to ascertain these large families and genotype them with 970,000 SNPs. And I'm reminding when I say this that when I first started, 300 markers took a whole year, and 970,000 SNPs can be typed in 3 days. So it's been a real change. So this is an example of a family that we recently ascertained with Perot syndrome, and it's a novel gene for Perot syndrome. And Rabia Fridi, who's a postdoc in the lab, is working in collaboration with a group in Bill Newman's lab in Manchester. At any rate, we do exome sequencing, and we first focus on the genes in the linkage interval that's been identified, but we, of course, evaluate the entire exome. And then we engineer animal models, mostly mouse, although we made one zebrafish model in Katie Kin's lab. And I'm very interested in fruit fly models, and actually my PhD was using fruit flies. So and it's a very good model. And of course, the question is, does the animal model recapitulate the human deafness? And usually it does, but I'll give you an example of where it doesn't. We then, if it does recapitulate it, we do biochemical, biophysical, and cell biological methods to understand the function of these genes and think about therapeutic opportunities. And so this is one project that we're working on right now. A lot of people have been working on the tips of stereocilia where the mechanoelectrical transduction channel is located, but we prefer the base of stereocilia where the rootlets are located. And a number of genes that we've identified and also identified in Peter Gillespie's lab are important at the base of stereocilia, where the stereocilia have to pivot. And without the rootlet, the stereocilia just flop over. And this is work being done by Ina Belyansova in collaboration with Bo and Chang. And then another gene that we're working on at the base of stereocilia is trio BP1 and 5 and anchored RD24. But what I want to focus on for the remainder of the talk is on TBC1D24. So we started working on TBC1D24 when we identified a very large family segregating non-syndromic deafness, and talked with my friend Suzanne Leal, who we publish a lot of work with. Does she have any families? And she did. And this was all published together. So I want to start off by saying that TBC containing proteins are abundant too in the mammalian genome, but there's only one with a TLDC domain. I'll come back to this point in just a little bit. Maybe you got to be stepping a little closer. Okay, so TBC1D24 is the only TBC containing protein with a TLDC domain, and this is an AlphaFold model. It has two globular domains. What's been reported at about the time we published our observation is that TBC1D24 interacts with ARF6 to modulate neuronal projections. I don't think the interaction is direct, but it was reported as direct. We did immunoelectron microscopy. Ron Petrelli, who's now retired, and TBC1D24 on a validated antibody by immunogold electron microscopy is located near the synapse, and I'll come back to this in a little bit. So one of the most interesting set of observations that have been reported about TBC1D24 come from this ortholog called Sky or Skywalker. Some of the alleles are embryonic lethals, but others allow the larva to survive for a while, and it sticks its head up and eventually dies, but it's because of increased neurotransmitter release causing paralysis and death, which is kind of consistent with our immunogold localization. Oh, okay, there we go. So there's been a fair amount of work done on other proteins. There are 5 other proteins that have TLDC domains, and OXR1 reduces oxidative stress in neuronal cells, and when it's knocked out, neuronal cells cannot suffer the stress of they die from oxidative stress. And it's also been reported that Sky in Drosophila is an oxygen sensor that mediates synaptic vesicle trafficking. But there's no evidence that TBC1D24 has anything to do with oxidative stress. Wouldn't surprise me if it did, but there's no evidence for it at the moment. So the top family, the large family, was one that we ascertained in Pakistan, and the other 3 families, Suzanne Leal had also ascertained in Pakistan with her collaborator. And there were 2 different mutant alleles in these 4 families. They were amino acid substitutions. And then a while later, Risa Tona, who was a surgeon in Japan and did her PhD in my lab, reported this family, and it has deafness and seizures in some of the individuals. And if you remember the title of the paper that we published, it indicated that we already knew that mutant alleles of this gene cause epilepsy. So now there were families with deafness and epilepsy, and many more have been reported. So I'm going to unpack this slide, and I don't expect you to follow it, but I want you to notice that within a few years, a slew of mutant alleles have been published for this gene. They cause deafness and seizures, and Door syndrome. And so I want to unpack this a little bit now. So the disorders are both recessively inherited and dominantly inherited deafness. DFND86, which we reported, and then Richard Smith's group reported DFNA65, and the same allele was reported in a large Chinese family. And numerous variants have been associated with epilepsy, from really severe epilepsy, and I'll tell you about an example of that, to just mild epilepsy, relandic epilepsy, exercise-induced epilepsy. And then there's DORS, which is deafness, oncodystrophy, and I'll show you some examples, osteodystrophy, intellectual disability, and seizures. So you might think that these different clinical phenotypes would be associated with different parts of the protein, or at least in different parts of the gene, and that's not the case. So, and I'm just going to give you a few examples as I unpack that complicated figure. So there's only one, there are two now dominant alleles. The one that's S178L is a dominant allele. The others are recessive, and they're distributed across the entire gene and across in each of the major domains, and including in the linker region between these two domains. The mutant alleles can be homozygous or in compound heterozygous condition. So the same is true for epilepsy. They're distributed across the protein, across the gene, and both the TBC domain and the TLDC domain, and in the linker region. And that's true for DORS as well. So that's perplexing. And this was a paper published about 10 years ago by Philippe Campoux, who's at Montreal. It's probably the best review of this disorder. And this just shows some of the children. And you'll probably notice that they either don't have nails or they have dysmorphic nails. And this is just a close-up from this Philippe's paper. So we started off by making mouse models because we were going to figure out what the cause of deafness is in the inner ear. So we made several, but I'll just tell you about two of them. We made the D70Y. This is a very highly conserved protein. It's 96% identical between mouse and human. So we made the D70Y, and we made the S178L, which is dominant, and either as a homozygous mutant allele or as a heterozygous, the S178L has normal hearing, and so does the D70Y. So that was a disappointment. So we cooked up some possible reasons why they would have normal hearing. One is there's a modifier in the genetic background, and we've had experience with modifiers that are dominant and can rescue hearing. DFNM1 is an example that we published. So that's possible. So we're putting these mutant alleles on a couple of different backgrounds, but we don't have the data yet. It's possible that the local structure is different, so that a D70Y in humans causes a disorder, the hearing loss, but it doesn't cause a hearing loss because of the local structure of the mouse. But again, it's 96% similar, and I doubt that that's the explanation. The other possibility is that TBC1D24 in the mouse is just dispensable. You don't need TBC1D24. One of those other 41 TBC-containing proteins and or the other 5 TLDC-containing proteins compensate for the loss of TBC1D24. So the next thing that we did, and this is work of Isabelle Roux in the lab, is we deleted exon 3. And it's a embryonic lethal. So we know it is it's an important gene for development in mouse. What we haven't finished doing is conditionally knocking it out in the ear, and that's underway. So we can eliminate this idea that the loss of TBC1D24 function is tolerated from a protective paralog or ortholog. So the next topic I want to talk about is some new data on a small molecule therapy for one of these mutant alleles that we've made in the mouse that is absolutely identical to the mutant allele in a human family. And these are the individuals. Yasuko is a surgeon from Japan. Wei Chen is my colleague. Liz Wilson works in my lab and does INA and Sayaka. So the mutant allele is S324 threonine frame shift as a result of 2 nucleotide deletions, resulting in a stop codon downstream. And it's a mutant allele in this linker region. And these are 2 of the papers that describe this family that has this mutation. And as you can see, there are 4 of them that have died already from disorder. So the mutant allele is located in a microexon. And the microexon encodes 5 amino acids. And it's highly conserved. The nucleotide sequence is highly conserved in mouse and rat and human, and the amino acid sequence is conserved, and you would think it has a very important function. But again, we were surprised. So Risatona made this very same mouse, deleting a CT, and resulting in a TAA stop codon just downstream. And the mouse, I can't show a live or dead mouse. It's not allowed to. So we made some drawings. So this mouse has wild running, convulsions and post-ictal state. Sometimes they live, sometimes they die, but they don't live for very long. So each dot is the position of the mouse, and the color is the speed at which the mouse is running around in the cage. And what you can see on your right side is the homozygous mice. They race around the cage. Not all the time, but when they start having a seizure, they do this wild running. The heterozygotes there might be a small heterozygous effect, but the wild type is clearly behaving normally. So the seizure onset begins at about day 14, 2 weeks after birth. And by day 21, 22, virtually every one of these homozygous mice have seizures. And they are dead by 3 weeks, every one of them. So the homozygous mutants are in red. There may be, again, a heterozygous effect a little bit, and then homozygous wild type are in the dark blue. So thinking up a therapy for this, one of the first questions, which I'll tell you about, but in a minute, one of the questions we asked is this microexon necessary? Because there's alternative splicing of it. It can either be included or not, and the downstream reading frame is the same whether it's included or not. So we floxed the microexon and we deleted it. And given its conserved amino acid sequence, we thought it would be very important for some function. So we made 25 matings between heterozygotes for this mutant allele. And in nearly 200 mice, we got almost a perfect 1:2:1 ratio, Mendelian ratio. So they're born in the expected ratios. And in addition, males and females are fertile. There are no seizures. They have normal hearing. And a mouse pathologist at the NIH examined 50 tissues and can find no apparent abnormality. So we don't know what the function of the transcript is with this microexon, but we're starting to do behavioral testing. And you'll see why in just a moment. So it turns out that the reason why this mouse lives is because this transcript that includes exon 4 doesn't appear in the brain until after birth. The majority of the transcript in the brain doesn't include it. So it's as if the mutant allele is absent. And the onset of the inclusion or the isoform switch parallels almost exactly the decline in the viability. So the survival goes way down as this transcript that includes this frameshift mutation appears. Okay, so the next thing we did is to ask whether we could interfere with this splicing to include the microexon using a small antisense oligonucleotide. And a lot of this work was pioneered by Michelle Hastings and others, and this is one of her reviews from nucleic acid research. That's an image. So the ASOs get into cells very well. They will bind by base pairing, and they can inhibit or activate splicing depending on how they're designed. So there are also a number of FDA-approved therapies using ASO, and I'll just quickly list. You'll notice there's a lot of Duchenne's muscular dystrophy, spinal muscular atrophy. They work very well. These bases are modified by putting a sulfur instead of an oxygen and a methyl group instead of a hydroxyl group. And what that does is it makes these nucleic acids quite resistant to RNases, nucleases. They have improved retention. They get into cells more easily. And the really surprising thing is that you can look a year later, 6 months later, and still find them in cells. And the way you find them is because Ionis Pharmaceutical has made a pan antibody that recognizes these modifications. And the antibody works very well. They're well tolerated, and because of base pairing, you can make them very specific. So can we cause the skipping of the microexon that includes the frameshift and the stop codon and prevent seizures in those mice? And just to reiterate, what we want to have happen is for the splice acceptor site of exon 3, the splice to the splice donor site of exon 5 and skip the microexon. And so Ionis designed quite a large number of these ASOs for us, and I'm just showing you the results of 2. So the upper line is the transcript with the microexon, and the lower band is the transcript without. And what we want is to see more of the lower band and less of the upper band. And it's over these 2 concentration ranges. And we did find 2 ASOs that pushed the splicing towards the exclusion of the microexon. So Yasuko Ishibashi, who's a surgeon, started injecting about 50 micrograms ICV into P0 to P4 mice. She was blind to the genotype. 3 other people genotyped these mice, 2 by PCR and 1 by sequencing, because we had to know for certain if a mouse survived that it was a homozygous mutant. And so this is the result that we have at the moment. The untreated, of course, are dying quite rapidly from seizures. The wild type and the heterozygotes are surviving, so there's no detrimental consequence. But we've gotten very long survival of at least some of these mice, up to about 180 days, from one injection between P0 and P4. So I think we can conclude from this part that lifespan of the TBC1D24 serine 324 threonine frameshift homozygous mutant mice can be extended to T1 182 days after birth. So obviously we're going to try a second injection or nasal inhalation or even a peritoneal injection to see if we can extend the lifespan to up beyond the 180 days. So I want to come back to this clinical heterogeneity because it's been really perplexing. How mutant alleles distributed across this gene, distributed across the protein, could cause so many different phenotypes. And so there was a recent paper, I love this quote now, proteins rarely act alone. So the idea here is that the clinical phenotype associated with the missense variants are the result, in part, could be genetic background too, in part from the binding impediments of different protein partners. So if we identify a protein partner, so what if we can't demonstrate that it's physiologically relevant? So I want to tell you how we identify partners and why we think the one that we started working on is relevant. So we started looking at partners by doing yeast-2 hybrid screens. And just to tell, for those of you who are not familiar, in a yeast-2 hybrid screen, there's a bait. In this case, it's going to be TBC1D24, and there's a prey. And since we don't know what it is, you're going to screen a library. And the idea is that if the two interact, you're going to get live yeast. And that's because the bait has a DNA binding domain associated with the protein in frame. And the prey has an activation domain. And when the two are brought together, you transcribe genes that are necessary for yeast growth. So the bait and the prey, if they don't interact, then there's no growth. So we found we don't have prey libraries. We could make them. We've made some, but we didn't have prey libraries. So we asked Hybridgenics, which is a group in France, to use the mouse TBC1D24 and screen some libraries that they had for interacting partners. They're a great group to work with, by the way. So they identified C2 domain of Kibra as an interacting partner of TBC1D24. And what I discovered is that there are hundreds of papers on Kibra. I've got a pile on my desk about 4 feet high. And they've been published over 20 years, and they're still being published. So it modulates synaptic plasticity. It's associated with episodic memory retrieval ability. There are SNPs in this gene that improve or lessen retrieval of episodic memory. De novo variants are thought to be involved in Tourette syndrome. It's involved in the hippo pathway. It's necessary for exosome secretion in certain cell types. There's a recent paper in Science which says that it's been positively selected for evolution of humans, along with 7 or 8 other genes. And there are 20 different proteins that interact with Kibra. So it's a protein hub. But no protein had been shown to interact with its C2 domain. So the first thing you have to do is validate this. And so that's what Sayaka did. we had the yeast-2 hybrid working in the lab, and these are the controls. You have only the vector, the bait vector, the prey vector, and when they're present in a yeast cell, they will allow growth on this, what's called 2 dropout media. But if you have 3 items dropped out of the media, the only way they live is if the 2 proteins, the bait and the prey, interact. And the 3-AT is called 3-aminotriazole, and when you add that to the media at high concentration, the interaction has to be very strong. And what you'll notice is TBC1D24 full length, this is mouse, and mouse Kibra full length interact. And I'm going to just tell you about the mouse data, but the exact same data is true for human, and we did all of those experiments. So the next question is, can we validate this? Oh, that's just showing they interact. The next question is, can we validate this interaction by another assay? And for this, we used an assay that we invented and published in 2017 called a nano-SPD, nanoscale pull down. And we've made it much easier than what I'm going to tell you about, but this is the basic feature of it. So a myosin 10 motor with its tail chopped off will move out to the tips of filopodia in a HeLa cell and a HEC293 cell. And we added on to the tail of myosin 10 a nanobody, a small antibody that recognizes GFP. So if there's a bait present and the myosin 10 motor and the EGFP bait, these are 2 expression vectors, are in the same cell, you'll see green filopodia tips. And if you also transfect the prey with an M cherry, the nanobody will not interact with M cherry, and the M cherry doesn't prey doesn't go out on its own as a control. What you'll see is this yellow signal at the tips of filopodia. So it can be done very quickly because you can buy GFP and M cherry expression vectors for just about anything. And you can transfect them into a cell, and you don't need a confocal microscope. You can do this with a fluorescent microscope. So when we did that, and you can quantify it, but when you did this, when you have EGFP with TBC1D24 and the M cherry Kibra, and I'll show you just at high magnification, you get yellow tips of filopodia, whereas if you have no bait, you just get green. Okay, so, and we also did co-IPs, standard co-IPs, and get the same result. So the next question is, what does the C2 domain of Kibra interact with in the TBC1D24 protein? Does it interact with the TBC domain or the TLDC domain, or maybe the spacer? And so we did those experiments by using as bait full-length TBC1D24 now as a positive control, TBC domain alone, TLDC domain alone, the mouse Kibra full length, and the mouse Kibra C2. And what you'll see is that there's no growth of Kibra as the prey interacting with TBC domain of TBC1D24 as the bait, but it does interact with the TLDC domain. And we've again done this for humans, and we've done this for mouse. And in addition, Sayaka has made lots of other missense mutations. But what we then started with to try to see if it's physiologically relevant is to see if any of these missense mutations, which are kind of around the periphery of the TBC domain, interfere with the interaction of Kibra with the TBC domain, with the TLDC domain, excuse me. So, again, they're almost identical, these 2 proteins. They're 96% similar. So we first did the mouse and then the human. And the box 6 mutations are sort of around this globular predicted structure for the TLDC domain. And what we found is that one of the variants in the mouse, A517 valine interferes with the interaction with Kibra. And none of the others did, except one, which I'll briefly tell you about. So this variant, which is A515 valine, alanine to valine, was reported in this paper by Ballestrini in 2016, and there are 8 affected individuals, and it's severe epilepsy. Yeah, almost at the end. Okay. So another question that we asked is TBC1D24 and Kibra expressed in the same cell? And we don't know that the source of the epilepsy is the hippocampus, but we're working with a group who does electrophysiology to figure that out. But what you can see at low magnification is that TBC1D24 and Kibra are generally in the same regions, but they could be in cells that are next to each other. And so Yasuko looked at single cells, and they are both expressed in the same cell. And you can look at RNA-seq data, and you can see that single cell RNA-seq data, and you can see that they're both expressed in the same cell. So the summary of this is that human and mouse TBC partners with TBC1D24 partners with Kibra. The partnering occurs between the C2 domain of Kibra and the TLDC domain. Both mRNAs are for these two proteins are expressed in the same cells in the hippocampus. And a variant that causes severe epilepsy interrupts the interaction. What I haven't shown you is that a second variant of that causes mild epilepsy has a weak interaction with Kibra. So we've got 2 points: weak interaction, a mild epilepsy, an absence of an interaction, and significant epilepsy. And so that means that these two protein hubs are interacting probably at the synapse. So a lot more work has to be done. So for future questions, what are the other protein partners of TBC1D24? Do these amino acid substitutions disrupt interactions with the putative partners? Is there more than one interaction that must occur for there to be Doors syndrome? In other words, several partners fail to interact, and that's the more severe phenotype. How do these disruptions affect cell function? And of course, we want to know what will allow us to address what is not functioning properly in mutant alleles of TBC1D24 that cause deafness in humans. And so thankfully, there's no finish line, and that's what's so wonderful about science. And I just want to thank the organizers of the meeting for inviting me. I hope I didn't leave anybody off, but the organizing committees and all the people that I've interacted with so far. And then this is the group from my lab, although some have departed since this was taken last year. So, and then another person is gone, and then Takushi Miyoshi, who is an MD PhD, is just recently an assistant professor at Southern Illinois University. And then I want to also thank the collaborators on this project and many of the other projects in my lab, and especially the group at Ionis Pharmaceutical and Michelle Hastings, who's no longer in Chicago at the medical school, but has moved to the University of Michigan, and others. So thank you very much for listening. How much did that go over?
Do we have?
Any questions?
And we have online question from Dr. Kumar. Dr. Kumar, I will unmute you.
Hello, Kumar.
Hey, Tom, can you hear me?
Yep.
A very nice talk again, very intriguing. Just a quick question, perhaps I missed this. Do any of the individuals with mutation in KIBRA show hearing loss?
Really good question. So we have a KIBRA knockout mouse that we got from the Jackson lab. It has normal hearing.
I'm sorry?
The KIBRA knockout mouse that you can
Oh.
obtain from the Jackson lab has normal hearing.
Okay, just one quick technical question because I'm not familiar with this. With the ASO injection in the brain that you showed, was that a target domain that you were trying to reach with the injection? And if so, or if not, how far does the ASO sort of spread from where you inject?
Yeah, another great question, Kumar. So it's just injected in the ventricle and it spreads throughout the brain. And surprisingly, you can detect it all over the body within a few days. And 2 or 3 months later, with the antibody, it's still present, both in the brain and in tissues, wherever you look.
All right. Okay, thank you.
We got time for one more question?
Same question. I have the same question, Dr. Alagraman.
Okay.
Anyone?
Yeah.
So do you think that the treatment will be equally effective for the epilepsy and the hearing loss?
Well, that particular mutant allele doesn't have hearing loss in humans. But we don't have a mouse model that has hearing loss. And we've made now 5 or 6 different mutant alleles of TBC1D24 associated with hearing loss in human, and they all
hear
normal.
fine.
They all hear fine. Right. Or the genetic background is a possibility. It can make a huge difference. But we'll have to wait to see.
Okay, let's give another round of applause.